days. I think you've all been here before, so I don't need to give much of an introduction. We'll pass around the sign-in sheet. If you haven't signed it, please do so. This goes, it's pretty obvious, but there's no Tools and Tech seminar next week because of Thanksgiving. So we will, the next session or speaker will be the week after Thanksgiving. With that, I can just introduce our speaker. Today we have Andy Kahn, who's a grad student in DCMB.
Hi, so I'm Andy from Alexina's Fishke's lab, and so we do lots of computational proteomics, and today I'll be talking about a new database search tool called MSFragger that will enable us to really study lots of chemical and biological modifications in proteins. So just to give some background on shotgun proteomics. So in shotgun proteomics, at least, well, I guess there is only mass spec-based shotgun proteomics. We start off with some biological sample of some proteins, and we want to analyze it using a mass spec and get some sets of peptides from which we can identify proteins from. And the basic workflow, we start from sample, we digest into peptides, often using trypsin, and then we can deplete or enrich for certain things. So if we're interested in phosphorylation, we can do phospho enrichment, and then we separate out the sample using HPLC, and then we feed it into the mass spec. And obviously, fractionation is a very important part of sample preparation because the sample is just very, very complex, and the mass spec is not able to sample everything fast enough. So you can run your proteins on a gel and separate things out at the protein level before doing the digest, or you can digest and separate things out using simple columns, or you can separate things out using HPLC, and then you mix your samples again. So there's a lot of things that you can do with mass fractionation. And after you load your peptides onto the LC system, it gets separated over time. And at a particular time, those peptides are ionized by the mass spec, and they're measured by the mass spec. So this is a sample survey scan. So it's just a measure of what peptide ions are coming into the mass spec at a particular time. So we can stack the scan. So this is what it looks like. So this is very zoomed in. We're looking at a very narrow range of mass discharges, and this is in retention time. So this is from 33 minutes to around 37 minutes. That's around 650, and we'll proceed to 667. So it's relatively narrow, and I just want to show what the data looks like. So here we have a peptide ion coming off their chromatography. So at first you get a light signal, and it intensifies, and then it's done. So these are just peptides coming off of the chromatography, being ionized and measured by the mass spec. But just the mass of the peptide ion, we can't identify a peptide from that because you have all these isomers, and there's a limitation to how accurately we can measure the mass of these things. So in shotgun proteomics, we do something called data-dependent acquisition. So we tell the instrument to say, well, if you see something interesting, if you see some intense peptide ion, isolate it. We only want to look at that, and then we fragment it by hitting it with some inert gas so that peptide will fragment. And so it'll fragment along its backbone, and using current instruments, we mostly get YMB ions. So this is the workflow. So peptides come off liquid chromatography into the mass spec. The mass spec says, oh, this is interesting. Let's fragment it. It fragments it, and then we get a fragmentation spectrum. And from this fragmentation spectrum, we try to identify what the peptide is. So there are different strategies for doing this. First and the most popular is database search, where we go into the sequence database, and we do an in-silico digest of that database. We generate theoretical spectrum for all of these peptides, and then we try and compare these theoretical spectra to the tandem mass spectrum that we acquired. So if they're the same thing, then they should look alike. So there are also other methods which are less popular, but they offer a lot more flexibility. So in sequence tag-based approaches, the software will try to look for these differences in mass spectrum that corresponds to particular amino acids, and so you can then use that as a filter in your sequence database. So this allows for some potential, single amino acid variations and other things. Or you can go with de novo sequencing, where the software just tries to interpret the entire spectrum without a sequence database. But those methods are obviously much more noisy and computationally intensive than a simple database search, which has been around for probably 2.5 decades now. So so MSFragger is a database search tool, so I just want to give a bit more details as to how they work. So we have a measured peptide ion mass. Remember, the instrument, as it surveys all of these ions coming into the instrument, it knows the mass of that peptide ion, and then it fragments it, and we get that fragmentation spectrum. So the database search tool would take the measured peptide ion mass and use that as a filter when it accesses the sequence database and considers all these candidate peptides. So if I measure a mass of, say, 1,000, and the mass accuracy of the instrument is plus or minus 0.5, then I only select candidates from 999.5 to 1,000.5, and that really helps you narrow down what that peptide could be. And then it will try and correlate and compare and get some sort of score that represents how closely the two spectrum are alike, and then you can do some sort of statistics on it to tell you how good of a match that is. So there are lots of tools that does this. So Sequest, Mascot, they've been around for a very, very long time. They're proprietary, commercial, and then there's a lots of open source and free tools like Xtandem, Comet, MSGF+, MirrorMatch, OMSA, Andromeda, and then computers used to be slower, so this process used to take a long time. So there are people who try to optimize things. So pFind, Tide, Tempest that uses graphics cards, and they all differ in their scoring functions, how they compute their statistics, and how they're implemented. And they're all just matching these fragmentation spectra to these theoretical spectra, and they're all reasonably fast with modern computers. It takes minutes or tens of minutes per file, and in my opinion, they're all of similar quality. I know there are people who will strongly disagree and will say, okay, this tool is the best tool ever. But in my experience, they're all similar. In a large-scale experiment, they all give you pretty much the same number of identifications. That's a fixed error rate. They differ by a few percent. So, okay, seems reasonable. All these tools work pretty well for conventional workflows and behave similarly. So why did I develop another tool? Well, obviously, it's for unconventional workflows. So there's this thing called mass tolerant search. It's been around for a very long time, but people didn't really use it because the instruments weren't as accurate back in the days, and so the results that you were getting with mass tolerant search weren't that powerful. So in a mass spec experiment using these standard tools and databases, most of the spectra that you acquire remains unmatched. So it used to be really bad, where you only match maybe 10 or 20%, and now with these better instruments, we match a lot more, maybe on the order of 35, 40%. But most of the spectra we don't match. We don't know what they are. When we look at some of them, they look good, but we just don't know what they are. So in this paper, they did mass tolerant search. So instead of using that peptide ion mass to help us really filter down the list of peptide candidates, so the accuracy of the instrument may be 5 parts per million, we said, okay, well, let's pretty much not use that information, and let's use a window of 500 Daltons to allow for all these modifications or potential modifications. And the idea behind this is that even though we're matching these modified spectra to the unmodified theoretical spectrum, so lots of the fragments will remain the same, and this is how they match. And with high mass accuracy, the idea is that your noise of the background is sufficiently low that you can allow for this not very restrictive matching. And magically, this identifies 30 to 40% more spectra. So we're going from like 35% to 50, 55%. So this is pretty big. And we're observing all of these modified peptides that we normally don't consider or don't look for them. And now we're seeing all of them, common ones and lots of unknown ones. And it's simple, so you don't need all these specialized blind PTM tools. So there are tools out there that will look for these PTMs, but they don't really fit well in the context of these generic workflows where you have very good methods developed to estimate errors and all this other stuff. So, all of those specialized tools, you run it, it generates a specialized format, and you can't really do lots of analysis, and it's not really compatible with lots of different things. So this is very simple. You just change the setting in your favorite tool, and presto, you identify all these modified peptides. So we're excited, but we're not surprised that it works. I mean, it was done a long time ago, and we actually did something similar using another database search tool that I developed that uses a GPU, and we did something a bit more sophisticated with that, and you see a very similar profile when we compared it to a blind modification tool. So, and we do lots of large-scale reanalysis in our lab. So we're like, well, there's some questions that we wanted to ask. So are these modification rates preserved across different experiments? So common chemical artifacts like, oxidation, or or even biological ones like phosphorylation, are they similar in all these different experiments? Are there modifications that are unique to particular experiments? So we fired off our favorite search engines, and in our lab, we don't really use lots of the proprietary ones because they cost a lot of money, and we want to just farm it out on lots of Linux clusters or use Amazon Cloud or something. So we generally use Extendem and Comet. So we ran one file, and it took 100 CPU hours to analyze one run. And we want to look at thousands of runs. So, and in the paper they claimed, well, they only took 2 hours on their machine. So we ran it on one of the computers in the pathology proteomics facility, which actually has a proprietary software, and it took 11 hours. So they could have some, specialized modified version of the tool, but we don't have access to it, and we tried a whole bunch of different tools, and they all took a long time. So we were ready to shell the money for clustered cloud computes, but we started thinking, so what's the fundamental problem? So instead of comparing that one experimental spectrum to hundreds of theoretical spectra when we use that narrow filter, we're now comparing that one spectrum to potentially several 100,000 or millions theoretical spectra. So is there a better way of doing this? And the answer is yes. So for two spectra to be similar, they must share some common fragments. And we can index all of these theoretical spectra based on the masses of their fragment ions to quickly look things up that may be similar. So this is just some more details, common being within some mass tolerance, and we want to just index all of these fragment ions so that we can quickly retrieve their parent theoretical spectrum. And we ordered references within each of these bins so that they're ordered by their parent precursor mass. So we can use binary search to quickly narrow down the range of fragments that we're interested in. So this is the Degenerate workflow. Again, this is the standard database search approach. We digest the protein database, and then for each peptide, we generate a theoretical spectrum, and then we compare that theoretical spectrum against the experimental spectrum. And in the MSFragger workflow, we take the protein database and we generate a peptide index, and that's been done before. But we take this a step further and we generate this theoretical fragment index from that peptide index. And then we can simultaneously score one experimental spectrum against all theoretical spectra. And there's lots of efficiency that can be gained from doing this. So, whenever we develop a new tool, we have to make sure that the results that we get are correct. And we see that we compared MSFragger to Extendem to Comets to Sequest, and we're using the Sequest numbers that they published. And the numbers are a bit different from our FDR pipelines, but they're all pretty much the same. And then we also compared our results with ModA, a blind PTM tool, and we see fairly similar results. But performance-wise, when we do a mass-tolerant search, we're 150 times faster than these conventional tools, so Comet, Extendem. But for closed searches, where we're using that 5 ppm window, so there's not much computation going on. Even for normal conventional tools, it takes less than 2 minutes for that file. There's lots of overhead involved with just starting up MSFragger, so we take 24 seconds. slightly faster, but it's irrelevant. But for mass tolerant searches, we're going from 14 hours, 15 hours on quad core, 8 core workstation to 5 minutes. And you can imagine if we want to do this thousands and thousands of runs, it can make a huge difference. And we also show that the tool scales very well. There's a lot of optimization and software engineering that went into just letting it scale if you have a very beefy machine. And the reason why we gain all these performance benefits is that when in a conventional algorithm, when you compare a spectrum to a spectrum one at a time, that takes a few hundred operations. But in MSFragger, when we do a mass tolerant search, on average for each of these candidate peptides, 1.5. So instead of a few hundred things that we're comparing, we're only comparing 1.5. So that's why we get about over 100 times performance increase. And we also did try a bunch of different things. So now that we have our own engine, we can build in certain things to explore this strategy. So when we do a mass tolerant search, we don't recover all of the peptides that we get in a conventional search. So we thought, maybe there's lots of noise. Let's try boosting some of these unmodified peptides. But it turns out that doesn't really help. And there's a very good explanation for this, but I'm saving that for the bistro talk later in the afternoon. And for further modifications, when we do a mass tolerance or open search, you only get around 50% of the modified peptides. So you don't get perfect recovery, but we can also do the search with these variable modifications because we can do searching so quickly that we do gain, we get all of them back pretty much. And in modern mass specs, we often only get Y ions, and they're generated from the C-terminal end of the peptide. So as you can imagine, with this mass tolerant search strategy, we're matching these experimental spectrum to unmodified theoretical spectra. So in these cases, you may disrupt the entire Y ion series. So we added some complementary ions to help recover some of these peptides that may have a C-terminal modification. And we see that it benefits it slightly. So going back to original purpose of trying to look at these large-scale data sets with mass tolerant search. So we looked at the data from the paper that I just mentioned from Stephen Gee's lab. It's a HEC293 cell line with 1.1 million spectra from 24 runs. And then we also looked at some larger data sets. So some triple-negative breast cancer data, 20 million spectra, and some HeLa data around 3 million spectra. And we also wanted to look at some label data. So in proteomics, people like to do labeling for quantitation and comparing different samples. And there are some inherent challenges with analyzing that data using mass tolerant strategy, because to analyze that data, you also need to specify certain variable modifications. And that can increase the search time by, in this case for SciLac, 3 to 4 times longer. So you can imagine that if that one run takes 100 CPU hours, with label data, it may take 300 to 400 hours. So when we look at all of these modifications profiles, we estimate that it'll take 60,000 CPU hours to just analyze this data using conventional tools, and maybe 250,000 CPU hours for that 34 million spectra SciLac data sets. But using MSFragger, we can do this in a few days on a single workstation. And we also do some mass adjustment and calibration because we're pooling all of these data sets together. And the instrument calibration can be off by a tiny bit. We want to make sure that everything is standardized. So we have some calibration tool that before we calibrate the data, it's like this. You can't tell the amidation apart from the first isotopic peak, but afterwards, we can see 2 clear peaks here. So these are normalized modification rates. So the purple line is the HeLa, the brown line is the HEC293 data, and the green line is the triple-negative breast cancers. And we see that for common modifications such as loss of ammonia or oxidation and phosphorylation, there's a significant difference between these experiments. So the modification rates are very different, even though we don't expect them to be. So a lot of this is likely due to all of that sample preparation. So I mean, I wouldn't expect the phosphorylation states to be very different from these different samples, but yet they are different. So there is something happening during sample prep that alters how many of these things that we're able to recover. And then there are lots of other things that were just relatively unique to a particular data set. So for example, this was not annotated in Unimod, but we saw this mass 52.913, and just consulting a periodic table and seeing that it's associated with these charged amino acids, we thought, okay, it's likely to be iron. And then there are all these unknown cysteine modifications. So cysteine is just so reactive that we see in many cases unknown cysteine modifications. And then there are also these mass differences that are unique to a particular data set, but we don't really see evidence for that mass in the fragmentation spectrum. So those are also very interesting, and I'll talk more about them in the bistro talk in the afternoon. And we see that there's not much difference in the localization profiles of these different proteomes, and that's just to be expected. And from our SciLac data comparison, we compared the two different channels, the light and the heavy channel in SciLac data, with red being the data from the patient samples, and the blue is just a mix of different cell lines and in the super SciLac mix. And we see this distinct 30.011 in the patient samples that likely corresponds to this methyloadduct that's commonly found in FFPE samples. So MSFragger is packaged as a standalone Java program, and it uses all these common mass spec data formats, MGFs, MCXML, MZML, and writes PepXML. So it's compatible with a lot of different workflows. The peptide index is stored on disk. So after you run it with a set of parameters the first time, you'll save the peptide index, and the fragment index is just generated in memory when you run it, and it can be reused when you're searching multiple runs. So, and it will dynamically partition your fragment index to operate within how much memory you have. So if you're interested in doing some gigantic searches, that may take a few hundred gigabytes of memory, and you don't have that. MSFragger will say, okay, well, let me do this one chunk at first, and then I'll do the next chunk, and I'll do the next chunk, and it'll combine everything together at the end to give you your final results. So to run it, Java, jar, MSFragger, and then you give a parameter file, and then you give it a list of data files. And the configuration and the parameter file is very similar to the one that you would use for comments. And here I have a little video that will demonstrate what it does. So here, my directory. I have the program, I have some data files. I'm opening up the parameter files and just checking a few things. This is on a 28-core workstation, so so it doesn't have a peptide index, so it has to build one. So it's building the peptide index right now, sorting these peptides, merging, and then generating all these modified peptides. And then it writes the peptide index. And when it says operating on slice, that means it's generating the fragment index. It took 4 seconds, and now it's searching this file. It read it in 3.8 seconds. And here it will update every 5 seconds, telling you, how many spectra it's analyzed. So it's around 850 spectra a second right now. And for each of for each spectrum, it's comparing it to, probably close to 1 million candidates. So, when you see 1,000 spectra a second, it's doing 1 billion of these spectrum comparisons every second. And so it just goes in the next file. It doesn't need to rebuild the index because all of that is in memory. And it read the file in, it's searching. And so searching these files using conventional tools will take several hours on the same workstations. And here we see that it just takes a minute or so. Okay, so it goes to the next file, but told it to stop. And then we see the pep.xml file outputs, and we also see that pep index file where it stored the peptide index. And here it's just a pep.xml file. And when we run it again, now that we have the peptide index, it will skip off that generation and it'll just read in the peptide index. And you'll go straight to searching. So in conclusion, fragment indexing enables these super fast database searches, and it's 150 times faster than a lot of these conventional tools. There are no heuristics or simplifications, and what we're doing here is just a more efficient data structure. And so this allows you to do large-scale identification for all these, potentially unknown modifications, and you get close to 30% more PSMs in a typical shotgun experiment, and you can investigate lots of different things. The tool itself is cross-platform, it's Java and compatible with a whole bunch of different formats. And in this talk, I mainly went over the technical aspects of MSFragger, but we also did a lot of interesting applications using this tool, and we also applied the core algorithm to other things as well. And I'll be talking about that in my bistro talk later this afternoon. I'd like to thank the members of the lab, and I'll be happy to take any questions. All right.
If you put a new source database in there, will it check this? Will it notice that and make a new index?
Yes.
So do you have a sense from using this tool how many of the unidentified spectra you're getting that are due to unknown post-translational modifications versus maybe a non-tryptic fragment when it was supposed to be a tryptic digested sample?
So oftentimes when we see these non-tryptic fragments, so it's from in-source decay, so we get cleavage from the N-terminus, and that often results in a very negative mass. And these very negative masses, we do observe them in our profiles, but it likely accounts for maybe less than 5% of all these other things. So dominantly, most of the things have positive masses, and many of them are chemical modifications. We haven't been able to follow up on a lot of these because there are just so many.
Sure.
And we're just starting to scratch the surface of all of these chemical modifications. And previously in proteomics, I don't think people had the capability to look at this on a very large scale, and with this tool, we now can.
No, it seems like a fantastic idea. It obviously works and speeds it up. In terms of validating that it's just benchmarking against other algorithms, have you taken something like a phosphorylation enriched sample and made sure that all the modifications that you're detecting have been minus 80 Daltons or whatever the phosphorylation is?
So we do detect lots of phosphorylation events, but so the mass tolerance strategy is not perfect because it matches things to the unmodified theoretical spectrum. So you're going to lose lots of things unless you actually specify the modifications. So it's a relatively crude approach, but we still recover 30% more. And there are potential refinements to this strategy where you can add in the mass shift to the fragmentation spectrum, and you can modify the algorithm to do that. But we haven't done that yet, and we may get another 10, 20%. We don't know.
Talk a little bit more about your benchmarking there, because
Okay.
I'm sort of wondering how how do you have a couple of slides of that earlier on, right? So I guess do you have to have a data set where that has been analyzed? I mean, I guess it strikes me that it would be nice to have some sort of kind of gold standard data set, and that probably doesn't exist, or
for modifications?
yeah.
I don't think that exists. So
Yeah, I don't think so
yeah.
either. So
I mean, there are lots of exotic and rare modifications in there. So in their paper, they reported this modification that's only found in this one transcription factor, and there's this one particular mass. And when we look for this mass in the much bigger data sets that we looked at, we see it. And this mass only corresponds to this one particular peptide from that one particular transcription factor. And there are likely, thousands or tens of thousands of such instances that have never been documented before.
Sure.
And, we can start looking for some of these things now.
So when it's going to build the index based on the parameters that you specify in your config file, probably the mass tolerance. So I think 500 Daltons in the example you had.
So that's actually specified as search time. So the index is only dependent on your protein database and your digestion parameters. But after that's done, and obviously all of the variable modifications that you want to specify. But after that's done, the index is the same. So it uses the same index for a 500 Dalton search or a 5 ppm search.
Okay. Yeah. So that's not a factor. So it doesn't matter what you set the tolerance at. It's only indexing based on the actual fragment ion masses.
Yes. So when you want to look at 5 ppm, it just jumps to the right place and looks at the few fragments that's in there. Whereas in a 500 Dalton thing, you're looking through a much bigger slice of the index. Yeah, there's a lot that can be done with this, and we're just at the starting point of trying to understand what all of these things are, what all of these modifications are, and it's exciting, but we don't have the bandwidth to do all of that right now, obviously.
Sure. That's excellent. I'd be curious as to if so in a typical post-translational modified search of say phosphorylation again, you would specify they would look for peptides that have that mass modification on certain amino acids. But how many of those unidentified spectra are due to that same modification, but in some kind of weird fragmentation that is some different mass from that? I think that this type of thing would allow you to investigate, how many spectra I'm missing that are phosphorylated, but that don't have that canonical minus 80 Dalton mass.
Yeah, so we don't look for that on specific amino acids. So this is one of the things we looked at when we looked at these profiles. So oxidation, people generally specify that to be on methionine, but it's well known that it's also on tryptophan, tyrosine, and all these other things. But you see that here, but normally you just don't look for it.
Oh, I see. So that's what those are the amino acids along them.
Yeah. Okay. So phosphorylation is mostly on serine and.
Right.